Ion exchange chromatography is based on principle of attraction between oppositely charged molecule. It is widely used technique for separation and purification of proteins, peptides, nucleic acid, polynucleotides and other charged molecule, mainly because of its high resolving power and high capacity. In ion exchange chromatography, ions are electrostatically bound to a chemically inert and insoluble matrix. But unlike ligand, these bound ions are replaced by ions in the solution during separation process in the following manner. M plus A minus plus C minus will give M plus C minus plus A minus. It is obvious that M plus A minus is a complex of positive matrix bound to negative ion, and so only negative ion can be separated by replacement mechanism. Such process is called ion exchanger. Practically, ion exchanger are packed in columns for purification of given polyelectrolyte or protein. Protein is immobilized on an ion exchanger. Selection of ion exchanger should be principally based on pH and salt concentration. Protein bearing same charge will not bind due to repulsion in ion exchanger and will be eluted first from the column. Now, only desired protein will remain bound with the ion exchanger, and that can be washed off with another buffer of suitable pH and salt concentration that reduces the affinity between bound protein and ion exchanger. The pH of a buffer used should differ by at least 1 pH unit from isoelectric point of the compound to be separated. Usually, cation buffer such as tris, alkylamines, pyridine are used for anion exchanger, while anionic buffer like acetate, phosphate, barbituriate are used for cation exchanger. Different protein bind with ion exchanger with different affinities. The passage of a particular protein down through the chromatographic column is retarded due to interaction between protein molecule and the ion exchanger. The retardation is comparable to the rate of passage of solvent. The washing of a column is called elution, and by elution, protein with low binding affinity for ion exchanger will move down the column. Now we'll discuss about different type of ion exchange chromatography. There are broadly two types of ion exchange chromatography, that is cation exchanger and anion exchanger. Cation exchanger possess negatively charged group, and this will attract positively charged cations. These exchanger are also called acidic ion exchanger because the negative charge results from ionization of acidic groups. An ion exchanger are one having positively charged groups that will attract negatively charged anions. The term basic ion exchanger are also used to describe this exchanger, as positive charge generally results from association of proton with basic group. Table 1 shows charges exhibited by cation and anion exchanger during ion exchange chromatography. Now, based on functional group attached to matrices, they are further divided into two types: strong exchanger and weak exchanger. Strong exchanger are fully ionized at all normal working pH values and includes sulfonate and quaternary amines, whereas weak exchanger are ionized over only narrow range of pH values, and examples are carboxylate and diethyl ammonium. The examples of strong and weak exchanger are discussed in detail in Table 2. Matrices used include polystyrene, cellulose, agarose. Bonded phase ion exchanger are suitable for HPLC containing wide range of ionic groups and are commercially available. Porous variety are used on polystyrene, porous silica or hydrophilic polyethers and are particularly valuable for separation of proteins. They have a particle diameter of 5 to 25 mm. Most HPLC ion exchanger are stable up to 60 degree and separations are often carried out at this temperature owing to the fact that raised temperature. Now we'll discuss about different parameters which are important for selection of ion exchange support, and they are isoelectric point, ionic forms, porosity, particle size. First we'll discuss about what is isoelectric point. For amphoteric compounds, isoelectric point, that is pI of a compound at stability at various pH values, determines separation strategy. At a pH above its pI, the compound of interest will be negatively charged, and at a pH below its pI, compound will be positively charged. Thus, when compound is stable at a pH above its pI, an anion exchanger support is used. Cation exchanger support is used when compound is stable at a pH below its pI. The operating pH also determines the type of exchanger to use. Strong ion exchanger support maintains capacity over a wide pH range, while weak one loses capacity when the pH no longer matches an acid dissociation constant of its functional group. We'll discuss about ionic form. Most ion exchanger media are available in several ionic forms and may be converted from one form to another. Ionic form of a support refers to counter ions presently adsorbed to support functional group. Counter ions will exhibit specific selectivity for each support. The lower selectivity of counter ions towards support, more readily it is exchanged for another ion of like charge. Consequently, appropriate ionic form will depend on the relative selectivity of sample ion to be adsorbed. In general, ionic form should have lower selectivity for functional group than the sample ion, so that sample ion will displace counter ions and adsorb to the support. The sample ion can then be eluted by second counter ion with the higher selectivity for the support matrices. So in this way, ion forms influences the ion exchange chromatography. Now we'll discuss how porosity influences the process of chromatographic separation. Porosity of a support refers to total pore volume within the matrix of the support. Greater the pore volume, higher is the porosity. A very porous support may have either many small pores or a few large pores. Exclusion limit of support is identified by size of largest compound able to enter the pores under given set of the conditions. Porous material with high exclusion limits are recommended for high molecular weight compounds such as proteins, antibodies and other biomolecules. Low or high porosity medium with low exclusion limit are recommended for separation of low molecular weight compound such as inorganic ions and organic acids. Now we'll discuss about how particle size influences the chromatographic separation. Particle size is measured in micrometer with dry or wet mesh designation. The wet particle diameter will vary from support to support and depends upon the ionic form resulting from differences in the hydration of the particle. Smaller particle size provides higher resolution and typically require lower operational flow rate. Larger particle size yield lower resolution but can be operated at higher flow rate. Elution. Gradient elution is more common than isoelectric elution during ion exchange chromatography. Continuous or stepwise pH and ionic strength gradient may be employed, but continuous gradient tend to give better resolution with less peak trailing. Generally, with an anion exchanger, pH gradient decreases and ionic strength increases, whereas cation exchanger, both the pH and ionic gradient increases during the elution. So this is about how ion exchange chromatography technique operates. Now what are the applications of this technique in biotechnology? Ion exchange chromatography is widely used in separation and purification. For example, for deionization and softening of water, purification of solution to keep them ion-free, in biochemistry for separation of drugs and metabolites from blood, urine and other samples, and separation of organic mixture of acidic and basic compounds, and it is widely used for purification of proteins, nucleic acid and other charged molecules.